So this last section from chapter 25 is about the transport mechanism in plants. So we're talking about how they are actually moving the water and how they're moving the organic nutrients that they've made through photosynthesis. So recall that there's two types of vascular tissue, xylem and phloem. Xylem are the cells that move water and minerals, and phloem are the cells that move organic nutrients. So xylem moves water from roots to leaves, so it pulls up water from the roots and moves it up to the leaves, and they are sclerenchyma cells. Remember, they're very tough. They have secondary cell walls, and they provide structure to the cell. And xylem cells are dead at functional maturity. So when they're growing, obviously they're alive because they need a nucleus and they need everything in there to provide the information to make the cell. Once they reach maturity, they don't need the information anymore. Everything inside the cell dies, leaving just the secondary cell wall to provide the tube for water to move through. And also provide structure to the plant, like I said, because they're very tough, and so it helps the plant stand up. And it has two kinds, tracheids and vessel elements. Remember that tracheids are the ones that are kind of tapered at both ends. They're smaller. And then the vessel elements are flat. They are stacked on top of each other with pits at the end. That way water can move through. And the tracheids have pits in them as well to allow the water to move through. Then we have phloem, which moves the organic nutrients that the plant has made. And there are two types of phloem cells, which are sieve tube members and companion cells. And phloem cells are alive for the entirety of their life. But remember that sieve tube members lose all of their organelles because they are the tubes through which organic nutrients are moved. But they don't have a nucleus, they don't have anything like that. So they need to rely on the companion cell to be in charge of their cell. So they are connected through little holes called plasmodesmata, and through this plasmodesmata, their cytoplasms are connected. So basically the companion cell is running both of the cells. So the materials that move through xylem and phloem are mostly water. So we call them xylem sap and phloem sap. So even though xylem isn't all water, because there's a lot of minerals in it, and even though phloem, the purpose is to move organic nutrients, it still moves through water. And we know that water moves from areas of high water potential to low water potential. So we can use this to predict and understand how xylem and phloem are structured and how they move their nutrients through them. And remember that water has special properties due to its hydrogen bonding, that it's very polar. So that's what allows the water molecules to fill up the xylem cells, is their polarity. They climb up the side of the xylem cells through adhesion, and they stick to each other through cohesion. So let's talk about water transport really quickly. So here's a figure from earlier in the chapter, but we know that water moves inward. Sorry. Nope, that's not it. There it is. Okay, water moves inward from the root hairs, and this is the epidermal cells. Then it moves through the cortex, and then it reaches the Casparian strip, and so it has to go inside of the cells now. It has to cross a cell wall, go into the cytoplasm of a cell. Out here, it can move through the outside of the cells, but at the Casparian strip, it has to move inside. And then inside we have the pericycle, and then finally the vascular tissue. And so the water moves into the xylem cells that way. And it's an open system, so that's why we can predict it using water pressure. And so as water enters the roots, it creates positive pressure. It's kind of like pushing on it, call this root pressure. And root pressure pushes the xylem sap upward. So the water moves in because it's moving from high water pressure to low water pressure, moves in, and it's pushing everything in the xylem sap upward. And this isn't that's not what's pulling the water inside of the cell, but it's just this additional side effect, and it produces something called guttation. There we go. Guttation is when the xylem sap gets pushed up and kind of squeezes out at the very top end through the stomata. So all these little dewdrops right here, they're not actually dew, they're coming from inside the plant due to guttation. So make sure you know what that is. Then we get into how water actually moves through the xylem, which is called the cohesion tension model. And this is suggested by a passive xylem transport. So this is not active transport, it's passive transport. And so we know, like I said, that water molecules tend to cling together because of their polarity. And so the polarity allows them to interact with the molecules of the vessel walls. So they will stick to each other through adhesion, and they will through cohesion, they will stick to the walls through adhesion. And so they kind of like climb up the walls, and the water column, so the water that's inside, all these right here, this is the water column right here, they move upward due to transpiration. So since they stick to each other and stick to the vessel walls, when the stomata are open and a water molecule evaporates, they just all move up and follow. So it's kind of like it's a line. Like you guys are waiting to get your food at the cafeteria, and you're in this big long line, and as each person gets their food and leaves, you move up one by one. So as a water molecule evaporates through transpiration, the rest of the water moves up and it pulls everything behind it. So the column must be continuous. So if there's a gap, then the water it's not going to keep going. So let's say that when you're in line at the cafeteria, you're all like linking arms, because that'd be really cute. You're linking arms in line, and so you're pulling each other along. But if someone leaves, now you're not linking arms with the person next to you anymore, so as you move up, you're not pulling the person with you anymore. So that's why a gap causes problems. That's why when you cut a flower, you have to get it into water as quickly as possible, because you just put a hole in the water column inside the xylem. And then there's the waxy cuticle, which is on the end of epidermal cells, which helps prevent water loss. So that's the cohesion tension model. So cohesion is that they stick to each other, and tension is that as one leaves, they get pulled up. So that's the cohesion tension model of water moving through the xylem. There's that image again. So make sure you read all of this. Basically, tension pulls the water column upward from roots to the leaves. There's water molecules, and as they leave, another one moves into place, getting ready to go. Another analogy would be like the batting order for baseball. So one person's batting, another person's on deck. So let's say the batting person is no longer in the queue. So they have left. They have evaporated. And the next person waiting right here is on deck. And then as soon as they're free to go, they leave, and then we move up in the batting order. So that's another way to think about it. Then you also have to know about the stomata. And remember that the stomata are cells that are in the epidermal tissue, usually on the underside of leaves. And they're two cells, two guard cells. And so these guard cells either fill up with water or lose their water, and it regulates whether or not the stomata are open. So as water enters the guard cells, it raises the turgor pressure, and so that's basically the pressure that water is having from the inside on the cell walls. So as the water moves in, it raises the turgor pressure, and the shape of the cell changes. Now the guard cells become full, they become turgor, they become turgid, and they open up and open the stomata. So right here, we are filled with water. Our vacuoles are filled with water, and the stomata have opened. When water leaves, the turgor pressure falls and the stomata close. So here you can see the vacuoles are much smaller, and so they're no longer pushing outward on the cell, so the cells just relax, and they do not, now here, when the stomata are closed, they do not allow any water in or out. And the way that this water is regulated moving in and moving out of the guard cells is by active transport of potassium ions, K+ ions. Because water will follow the K+, because it's going to move from high water pressure to low, or high water potential to low water potential. So if the cell wants to open the stomata, they are going to pump in potassium ions inside of the cell. So active transport, they're going to use ATP to move those potassium ions inside of the vacuoles. You can see all of them in here in this figure. Then water will follow. So we use active transport of potassium ions, and water follows by passive osmosis. Then when we're done, when we want to close the stomata, we pump all of that potassium back out, the water follows, and it leaves. And now the cells are flaccid and they close. And there's a bunch of things that regulate when stomata open or close. Things such as blue light, if you guys read that, the plants can detect blue light, blue-green light, and so from the sun, the blue light from the sun regulates whether or not the stomata are going to be open or closed. Makes sense, they want to be open during the day and closed at night. That way they're open when they're doing photosynthesis and closed when they're not. Then there's also an internal biological clock. If you keep the plants in the dark, they're still going to regulate their stomata opening and closing for a while until they finally realize they're in the dark. And then temperature, humidity, and stress also regulate opening and closing of stomata. Now let's talk about how things move through the phloem. So basically we have transporting of sugars and other nutrients. So we transport sugar and other nutrients from places that make sugars. So our photosynthesizing tissues. So these are things like basically anything green on the plant. The leaves, if the stem does photosynthesis and it's green, those are going to be places where they're making sugars, and we call those sources. They are sugar sources. Places that are making sugar are the sources. And then we move these sugars to places that need the sugars, places that they do not make their own nutrients. So this would be things like young leaves that are still growing, and so they haven't maximized their photosynthetic potential. Things like flowers. Flowers do not do photosynthesis, so they need the sugars from someplace else. And roots obviously are below the ground, so they don't do photosynthesis. So those are the three kinds of sinks. That's what we call them. So it's kind of like you're pouring sugar into the sink. So sources make sugar, sinks use sugar. So we use the phloem to move sugars from sources to sinks. And there's a little example about how they found out that bark is involved in movement of organic nutrients. Read that about girdling. And then the model that we use is called the pressure flow model of phloem transport. And so basically, positive pressure drives the sap in the sieve tube members. So sieve tube members are where we move sugars through. Remember, the companion cells are on the outside, regulating everything inside, because there's nothing inside of the sieve tube members, even though they are alive. So we actively pump sucrose, a sugar, our organic nutrients, into the sieve tube members. And then water immediately follows, just like with the potassium ions. And then this increase in volume creates flow that moves water and sucrose to a sink. So if this is a sink up here and this is a source, source, sink, green is the source, the roots are the sink. Up here, these guys are making their sugars and then they pump them inside. And then as soon as they pump the sugars inside, the water follows, and now there's a big buildup of nutrients up at the top right here because there's a lot of water and a lot of nutrients. And so just makes sense. They're going to move down into the empty spaces right here, and so they just get pumped and pushed into the roots or into whatever sink we're talking about. And that is the end.